Are a little bit of housekeeping. What I did send to you that little bit of additional study guide materials. That's for. There's only a couple little items in the start of the cell, and I need them to build on everything else. And interestingly, so we're basically looking at membrane proteins and what they call cell adhesion molecules, the way cells link together. Kind of some of the stuff you might have learned about with epithelial tissues in lab. Nevertheless, because that sort of sets the framework for really the next big topic, which is movement across membranes. And that's not on the exam, but in covering movement across membranes, it should reinforce the activities of those membrane proteins so you'd be more familiar with them. So I think they'll sort of dovetail nicely together, so to speak. That's one. I just emailed you, and what I found out, when you have these large files that have a lot of animations and things, they're apparently too big for. We made the transition completely over the summer that we started last year to this Blackboard Ultra. It's a little bit easier, and it's more geared to doing things that are ADA compatible, which businesses and universities have to do. It has some nice wrinkles on that particular Blackboard. Hopefully you're getting some use out of it with other teachers with discussion boards and presenting things and stuff like that. It's just not my style. But nevertheless, the reason I have to email them to you is too large. Anything over like 100 megabytes, they just spit it back at me. So look for those, please. You can open them separately, or you can just look at the recordings when you're reviewing the material. You're certainly under no compulsion to download anything. Okay, all of that good stuff. And I have a little supplemental outline here that goes over the parts of the cells. You can open it at your leisure. Okay, and that's really the business part of it. So we're going to finish up now with what we had started, and finally finishing this biochemistry chapter, which is we're going to do the nucleic acids today, which are DNA, RNA, and they are the polymers. So just like proteins, okay, there's monomers and polymers. They're similar. In proteins, everything was an amino acid. That was the monomer. The distinguishing point is there were 20 different ones. Thankfully, you don't have to know other than positive charge, negative charge, acid, basic, etc., like that. Nucleic acids are similar in the fact that they're constructed similarly, and there's really only 4 of them. And so, but because things like RNA and DNA really have no length limitations, you can just imagine all the possibilities. Even though there's only effectively, rather than having 20 letters in the alphabet, you only have 4 letters in the alphabet. And that's really the similarity. The other similarity that's noteworthy is this. These, unlike fats and carbohydrates, these have nitrogen. So amino acids by definition have nitrogen, and indeed what you'll see is the makeup for all of these, for any of the nucleotides, the basic building block involved effectively what's called the nitrogenous base. Our bodies have a very limited tolerance for nitrogen. We ingest a lot of proteins. It isn't a big problem, and we have a very high protein diet. It's just given because we're not an impoverished country. Proteins, we have to deal something with the nitrogen. Your kidneys, probably the first job the kidney has more than anything else is getting rid of excessive nitrogen. We don't have a limited tolerance. So amino acids and nucleotides and nucleic acids, all of those are the nitrogen-containing compounds, and we do have some problems with the waste products. For instance, when I show you what's going on with the different classes, I'll tell you about a disease called the gout. Very, very briefly. I mentioned in passing with crystals, sharp crystals stick in the joints. It's because of an abundance of something called uric acid. That comes from this word, purine. So we can have some of it floating around in our bloodstream, but we can't have too much, or at least this disease called the gout, which is all about endocrine. I'm trying to give you I always try to give you some context. I mean, that's what I'm trying. So it's not, I said it without using the appropriate terminology to probably the first class. I'm sure you leave here sometimes and going, why in heaven's name are we having to learn all of this stuff? We're probably not saying that and using that same terminology. Some of you may be polite in saying it. But if I was sitting where you are, I go, why the is he telling us all this blank? Okay? The reality is, this is all the context to begin to understand how to apply this is coming. But we have to set the framework and the building block. So it's getting there. It's not something you have to memorize. It's something that's going to help you down the road. And I hope that's the reason I hope you have an appreciation for it. Nevertheless, we have a nitrogenous base. In this case, when it's a nucleotide, we have a 5-carbon sugar. The big deal with these building blocks, is it ribose or deoxyribose? That's absence of that oxygen allows the magic of DNA to occur because it's all about the structure. And then we also have a phosphate group, and we'll get to the importance of those, particularly when we look at the nucleotides, and particularly with the one that's focused on here called ATP, our energy currency. That's the frame we start with. So of those nucleotides, the small molecules, the monomers, we build 2 giant molecules. Nothing is bigger molecularly for us than DNA, followed closely by RNA. So we'll deal with the DNA first. What is it? 2 big jobs, the 2 most important jobs you can imagine. DNA basically is the roadmap for construction of proteins. It's the roadmap for all of our genetic bases. I commented on it last time. I was looking it up a little bit yesterday as I was looking, just doing some readings that I do. As I hear about diseases and things like that, I look up stuff. I mean, really, I thought it was maybe it's even less than 2% of variation in anybody's DNA in this room compared with anybody else. Tiny amounts that account for the differences between us. And I mean, it's just fascinating, but the magic of this molecule is that it contains effectively nearly each of our cells have all of this information that they can basically express in the formation or lack thereof, or the availability or lack thereof of certain proteins. That's that story. And the magic of it is, it's in, as you will see shortly, and you're probably already aware of, it's called an alpha double helix. It's one of those right-handed twists, but there are 2 of them that are sort of intertwined, and it's a spiral staircase. And the magic of them is the steps are the same width. So it's that sort of unique spiral staircase. It's not wider here or narrower here, and some of the illustrations might endeavor to show you. Okay, so, and because they are connected, as you will see, by hydrogen bonds, they can open and close. When they open, they can tell us what proteins to make. When they close, they can just contain that information without it being disturbed. When they open, they can split apart and actually make 2 new companion strands of DNA and magically make more cells. And basically, that's all we do all day. We make more cells. And the vast majority of the time, as you are already aware of, or will be again, something called mitosis, all about identical cells. It's the whole ballgame. That's the magic of this molecule, and that it can open and close like a biological zipper because of the hydrogen bonds. That's the other thing. Well, we spend time on that just talking about water because these are similar molecules in the sense that they behave the same way, with a bond that's loosely attached, but attached enough to give it structure, but loosely so they can change that or open up as needed. So we get to what it basically has. And really, the history of DNA is interesting. We knew that DNA was important back in the early 1900s. Right after World War I, medical scientists and doctors realized how important by this time they had already identified that DNA consisted mostly of these nitrogenous bases, and there were equal parts of adenine and something called thymine. The names that you have to know are down there, or what category they're in, and guanine. They knew about this. It was just they weren't, but they couldn't put the picture together. And guanine and cytosine, they were always in equal amounts, but they weren't necessarily. It wasn't like 50-50. It could be 20-80 or 80-20 or somewhere in between. It didn't matter. But we knew they were equal parts. And how that contributed to the structure was not clear. They were aware of deoxyribose. They were aware of phosphates. And what, as it turned out, the two categories are different structurally. The purines. So we always use the A's, the T's, the G's, and the C's. It's easier than me to keep writing them because my terrible handwriting. We have adenine and guanine are what are purines. And they, as you will see, have, I'll just draw little boxes and you'll see it better. They are two rings, two carbon rings. And within those rings is nitrogen, and hence the name nitrogen. Structurally, we use a lot of nitrogen. That whole connective tissue thing you're doing in the lab, all that ground substance stuff, those are nitrogenated carbohydrates. They're sugars with nitrogen because it makes them structurally sound. The shells on insects are made out of something called chitin. That's a nitrogenated carbohydrate. The rough stem of mushrooms, okay, that you can't really eat, you snap off like the shiitakes, chitin. Nitrogenated carbohydrates, structural material. C, big part of the C, I better be careful, this is the G. And so cytosine and thymine are called pyrimidines. And they only have one ring. And so, given what, if we knew about this twisted ladder with uniform rings, they could have found out the structure early on. And probably they would have, but unfortunately they were interrupted because almost immediately after World War I came World War II. And what scientists and doctors do at that point, whatever their country is, whatever sides they are supporting, they immediately go to work for them. So really, there was very little science and discovery during that period of time. Okay, and post World War II, roughly in 1948, a U.S. doctor and a British doctor, along with his associates in England, and the two main fellows you heard their names are Watson and Crick, began to study in earnest. Another doctor named Linus Pauling out in the West Coast at Caltech was doing the same thing. The British guys got it first. And they got it first because of a lady whose name I mentioned to you before, who really never got credit and should have, named Rosalind Franklin. They knew the components. They just couldn't put the picture together. She did what was called X-ray diffraction. And what that meant is she would X-ray groups of, they had the DNA, they were able to separate it and try to come up with a little amount and take an X-ray so you could see the structure. The trouble is opening and closing all the time. She figured out, if I freeze this, I'm not talking about throwing it in the freezer at home. She did use something like liquid nitrogen, froze the materials, X-rayed it, and magically she saw that the steps were uniform. And they comprised exactly, okay, 2 on one side and a 1 on the other, or a 1 on one side and a 2 on the other. And it dawned on her that this was the uniformity. And the story goes that she discovered this in the morning or overnight. And when she met her colleagues, wherever they were having lunch or just sitting around and talking, she mentioned it to them. And it's almost like the light bulbs went off over their heads. And they immediately went up and took Tinker Toy models. Basically, the original, you can see Google it and you'll see the original Watson Crick model, and basically constructed the molecule that afternoon. 5 years later, they get a Nobel Prize. She was never recognized, never credited. A sad, terrible, and even to a limited extent present today. Thankfully, it's improving of not recognizing the valuable contribution of women in science and medicine, which is I can't even think of it. Horrible isn't strong enough word. She eventually was recognized but never received an award because you can't receive it after you have died. Only if they award it to you and you die before the ceremony do you still get the award. And that's happened actually relatively recently in Nobel Prize. Nevertheless, that's the big deal about all of this. She figured it out. The whole idea of complementary base pairing, the A's and the T's, the G's and the C's, they just fit naturally together. And indeed, if we have a situation, you'll see this. There's always going to be an A and a T and a G and a C. And if all we get over here, we have an A and an A, we have an error in DNA. That's a fancy name that you've heard before. It's called a mutation. Mutation doesn't mean magically you're going to grow a third eye or an extra arm. That might be useful in sports. Okay, it doesn't. A mutation means one thing, an error in DNA. If it causes cancer, it's got another name. It's called a carcinogen. If it causes a birth defect, it has another name. All of these I'm giving you for context, called a teratogen. Mutations occur constantly. We make billions of cells every day by mitosis. It would be naive to think that they were all perfect copies. They are not. The vast, vast majority of those cells are either not made, or the mutations are what are called silent mutations because they don't impact the genetic material of the construction. Vast, vast amount. Mutation rate for our cells is something like probably an average between close to maybe 1 in 100 billion. Ridiculously. We have all these safeguards. That's something you talk about a little bit when we start talking about it in microbiology, which many of you will be taking down the road. So just to give you an idea of the importance of DNA, one little error could be something that's incompatible with life, or one little error, as it frequently is, doesn't make any difference at all. So it's all because of that rule. More on that down the road. So it looks like this. Not exactly, because you don't get the sense from something like that that the steps are all equal in width, but they are. 2 right-handed helices, okay? And they have what's called a backbone. So famously, and this is what helps to maintain the structure, have phosphate, sugar, and I just put base, phosphate, sugar, base, and the bases could be different. It doesn't matter what I'm just doing it. Here, you see little dots, hydrogen bonds, and here you have the opposite, base, sugar, phosphate, base, sugar, phosphate. And what keeps the base, sugar, phosphate, bond is not only do the sugars and phosphates bond to each other, they bond to the next one in line. And this extra bit of bonding creates an area we call backbone. It maintains the structural integrity. So the big variability is in the middle. And as you'll note, if you look closely, this one's pointing up, down. You'll see in one of these animations that I'm going to buzz through, I'm not going to play the whole thing. It's something that you learn about in molecular biology, which is the directionality, and we actually name they're named like 1 through 6. You don't have to know any of that, thankfully, but what it means in terms of context of DNA is one strand is upright, the other strand pointing down. The fancy word, the strands are identical, but anti-parallel. One up, one down. They're going in the same direction. Parallel doesn't mean like in geometry, 2 parallel lines in a transversal angle, right? Did geometry, right? You remember that stuff? Love that. Before I became a chemistry major, I was a math major. If you could make designer t-shirts, I would have had science nerd all over mine. Love that stuff. So the beauty of that is it doesn't matter. When it opens to make more, it doesn't matter which one of these we don't make it from one or the other. We make them from both. They're the same thing. One's upright, one's down. There's a little bit different way when we assemble it. The one that's going in the opposite direction as to where it's usually assembled does create some issues that your body is able to correct. On the other hand, since they're both the same, no matter whether one's up and one's down or the other way around, it doesn't matter which one we use to make the proteins. The message is the same. Doesn't matter whether you start at the top or start at the bottom, depending, as long as you're going in the correct direction. That's the beauty of this particular system. So there you can see consistently the NHs and the OHs that are there are able to bring this molecule together. So it will open and close. It is large. It is so large. DNA is so large, it can't leave the nucleus. As you will learn, it's the nucleus is not the only place we find DNA, and it can't leave the other place either. Okay? RNA can. So in order for DNA to express its message when it's making proteins, it has to rely on RNA, which is a huge molecule itself, but linear. It's this double helix that makes it too large. And so what happens is we have the makings of a similar molecule using that code system, those letters, the A, and you've heard this, the A's, the T's, and the G's, and the C's, basically create, well, really effectively the genetic code. The only difference is when we get to RNA is we change the sugar. The sugar becomes ribose, and we substitute almost two identical molecules. We substitute uracil, which is another pyrimidine with a U there, for thymine. As you will see molecularly when you take a look at their structure, there's one little side chain that's different. For whatever reason, that substitution is made. And when you hear about the genetic code, if you study that in future classes or have studied it before, when it's able to tell you what amino acids code for what, and it's all 3-letter codes. It's a 3-letter alphabet. As you will see, they use U in that. So, and you can just go, show me a map of the genetic code, and it will give it to you. It'll be AAU, UUU, UGC, GCC, all that type of stuff. What amino acids are codes for. You don't have to know any of that stuff. I've never seen a science test that anybody's ever given where they haven't given you that map. And I wouldn't give it to you anyway because I don't feel the need for that. Okay, so RNA is basically the link to protein synthesis. Effectively, this is what happens. It's got a fancy name. There are two aspects of it. One is called transcription, and one is called translation. They're aptly named because when you transcribe something, if this system that records my voice and gives you a letter form was perfect, it would be a true transcription. I do not know what they do in hospitals now in an operating room. As a surgeon, I haven't operated in a hospital for 25 years. And that was right now that's where the Penguins play because they built the new arena right on top of that old hospital. Nevertheless, we would get to the end of it, we would have a dictation system, either had a phone with a button you could push or had, basically effectively a tape recorder. And right after the procedure, which is when you're supposed to do it, because if your memory's fast, you describe exactly what you've done. It's very methodical. You're saying the same thing all the time and little subtle nuances. That is recorded. That recording is sent to a medical transcriptionist at that time. And that person, he or she, would transcribe it exactly as you were speaking. If you wasn't sure of the spelling, I would spell the names for them just like I do for you. Right where I got that habit. In the old days, before we had all this elaborate technology, if an officer, whoever was in charge of the office, a man or a woman, they would have a secretary who would come in and they would take shorthand. Exactly what was going to be said as they spoke it, and then it would be typed and given for a signature and then sent out by the old mail. My mom did that. My mom was a professional secretary. So that's the idea of what transcription is. You're using perhaps a different language, and that's really what's here. It's like a kind of scientific shorthand, okay, that is taking the language of DNA and turning it into the language of RNA, even though the languages are very similar. So that first link of making something called messenger RNA. Down the road, you'll see that it's not quite so simple. Our bodies process that messenger RNA, so it undergoes some interesting changes that too limit the likelihood of mutations. It's called messenger RNA processing. I'm mentioning it in passing. If you have me in micro and we talk about it, we'll talk about that in a little more detail. Okay, that's something that students who are taking genetics and cell and molecular biology learn all the time. So that messenger RNA is the next step. That holds the transcription. It can leave the nucleus through tiny little pores, as you'll see on a diagram you probably have heard of. It takes that to a location called a ribosome, and that ribosome is constructed of ribosomal RNA, there's a shock, but also with some proteins. And they function as a base. And the two of these sort of join together and form a platform by which yet another form of RNA, transfer RNA, which transfers or pulls amino acids out of the environment surrounding the ribosome, because they're all over the place, and they're soluble in water, and basically identifies them, pulls them, and takes them in the appropriate order to the ribosome to be assembled into a polypeptide or a protein. Period. And we call that process because effectively we're taking the language from DNA, transcribing it, and then putting it into action by assembling a polypeptide or a protein. We call that translation. Translating the DNA into its end product, a protein. So remember, transcription, translation. DNA to RNA is the transcription, RNA to making proteins, the translation, if you want to make it very simple. And a lot of the tests I have, you'll hear me say this to you, are what I call KISS. Keep it simple, stupid. When in doubt, pick the simple answer sometimes. But this looks like such a better answer. Don't let that fool you. Wrong answers in tests are called distractors. They're there for a reason, to distract you. Don't fall into the trap. That's what I used to do. I got you. You went to you thought you had the instructor, and they could see me mouthing, I got you, son. I never got Miss Congeniality. Miss America contest, they had things called beauty contests, probably politically incorrect. See, that's a culture references that is lost. So this is I'm not going to play this for you. It gives you an idea. So they are effectively the nucleic acids or the polymers. Here, look what you'll see here. Here are the bases. And as I move along, now you can see here you can see the adenine and the guanine, the 2-carbon. You don't have to know anything about it, don't have to write it. Cytosine, thymine, and uracil. Notice when they come along a little farther, look at the distinction here. You can see, and I can't move the cursor, if you look at the bottom on the right, uracil and thymine, if you look at really what's sort of the 10 o'clock position, that's the whole difference. There's an extra carbon chain. Why am I telling you all of this? The name that we give these, just the nitrogenous bases, sometimes what we call nucleosides. Fancy name. We use nucleoside analogs today very, very frequently still to do two things: as antiviral medications and as anti-cancer chemotherapy medications. Okay, one of the earliest treatments for breast cancer, after, I'm not talking about surgery or excision or minor or major mastectomies, was uracil. We took uracil, and in that same position where the thymine is the number 5, if you look over there in the periodic table, all those halogens, fluoride, chloride, bromine, on the far right, but not the helium column, the inert column, those are all toxins. They took a toxin called fluoride, the F, they stuck it in that number stuff. Easy for me to say. Stuck it in that number 5 position and made 5-fluorouracil. That was one of the earliest anti-metabolites. Because the cancer cells gobble it up and they die in the process. Effectively, you're giving them something they're ravens for. All they do is divide, and they gobble up every nutrient there. If we give them a poison nutrient, we hope they die sooner than the person dies. Okay. The other thing, adenine up there directly above it, they substituted a chemical with that. If you know somebody, maybe you've taken it, who had a really bad cold sore, they took a medicine called Valtrex. Very, very good antiviral. Okay, one of the better antivirals. Guess what? It's a substituted adenine. So that's the context. Down the road, many of you are going to be looking at pharmacology and pathophysiology courses. This is why when you walk out of here, oh, that actually made sense. I didn't know that. You may not realize it for a year or two, but it'll finally sink in, I hope. And then as you'll see here, they show you about the assembly. See how they number those points? They do this all the time. It goes, there's a directionality to the assembly and disassembly of RNA and DNA. And you don't have to know any of that. It's annoying. The pre-med students, that's something they're kind of stuck with having to know. And that's really the picture for nucleic acids. Okay? When we get to nucleotides themselves, by definition, nucleotides are small, not big. They have the same composition. But it's not just ability, it's like glucose. What do we do with that glucose? We turn it into energy currency. The energy currency is called ATP. Okay, basically it looks like this. It has adenine, aforementioned base, sugar is now ribose, and it has, in the form of ATP, has phosphate groups, 3 of them, triphosphate. Phosphate 1, squiggly line, high energy bond. Phosphate 2, squiggly line, high energy bond. 3, all because they have negative. The combination, that's what ATP stands for, adenosine triphosphate. We use every little piece of this biologically. We generally in the body, when you make molecules, even parts of those molecules contain activity. That's why it's the most, to me, the most magical molecules in the body. It's so much simpler than DNA, which is so complex. If you combine adenine with ribose, or you combine guanine with ribose, okay, they both have similar names. When you add a ribose to one of those two, I just would add. You would equal guanosine, which we're going to learn about. Here, adenosine. I don't know what you did if you got up this morning and you were tired, but what I've been doing for the last, 50 plus years, in the morning, I block my adenosine receptors, okay, with caffeine. Adenosine is a very potent chemical in your body, particularly in the nervous system. When it's active, it calms you, you relax. It has a very important role in pregnancy and in birth and delivery. When we block its activity, it tends to activate our systems. The actual definition of how caffeine works, it blocks adenosine receptors. What has caffeine? Caffeinated beverages, colas, Mountain Dew is just packed with it. If you're into that super caffeine, all those energy drinks, coffee, tea, chocolate. Okay? So that's what I do in my way every morning. So that's one of the roles immediately that are there. And as you will see, so here's adenine and ribose. This is how they're connected together. Adenosine, number 1. Number 2, if we have just one phosphate, if these magically disappeared inside of a cell, this is a mediator. This is a chemical that generates activity. Down the road, you're going to learn it's got a fancy name. It's called a second messenger. That's in the next chapter we're doing. And now you have the phosphates, these two. If we added up how many times we created ATP, we took ATP, locked this off, made ADP. And if we counted each of those molecules, all the ATPs and all the ADPs, we were constantly either lopping off a phosphate to release energy to do work and putting it together to store energy. And I've said this to you before, if we added up all those trillions of molecules every day, it would equal approximately what we weigh. If you weigh 150 or 130 or 120 pounds, you'd have 120 pounds that much. It boggles the mind how much we use. Why it has energy is the phosphates. Those little red squiggles are high energy bonds. Okay. the best analogy, and if you come up with a better one, tell me. I promise I'll use it. Kitchen magnets. Someone in this room has played with kitchen magnets. You've probably played with kitchen. Do you have a question?
Yeah, what is that?
Oh, good. Oh, you might have bad handwriting. Oh. Oh. Here, sugar. These are phosphates. My wife can't read much. I read her a note every morning about it. Because I don't know what in heaven's name you spell. She doesn't say it. That's that kind of relationship. And she goes, I don't know what the you told me in that, so I didn't get a chance to do it. And I know and so I hope you know that it didn't get done. I know just what to say to her. Yes, dear, you're perfectly right, dear. I was wrong. Which is why I have lived to this 74 plus years. Your dads have all learned that lesson. Okay. Trust me. Okay. Nevertheless, that's the magic of this. And the whole idea of the kitchen magnets is when you had that, if they were the same, if they're opposite charge, they'd slam together. They're negative, you put your fingers on it, play with them, and let them push apart. That's what you're doing. To hold them together, you had to add energy. That's a high energy bond. If you had 3 of them, you had a whole bunch of energy. This one will explode off. We use that energy to do work. Every time insulin is active, and every time you eat something, insulin is active, assuming you can make it. I mean, that's only if you were a type 1 diabetic where you would have that, not have that capability. What insulin effectively, one of the things insulin does is opens a portal to allow sugar, glucose, to enter your cell. Immediately, upon entering the cell, it picks up one of those phosphates. That energizes the glucose. Remember I told you with what enzymes do, they lower the activation energy. Part of the way that we break down glucose into energy is we put energy into it right away as it enters the cell. That changes it. It's called G6P. You don't have to know that. This is a process called glycolysis. It's the start of how we break down and generate ATPs for our usage intracellularly. It's a magical system, but it always means that there's always glucose outside the cell and glucose is used up inside the cell. It creates something that you're going to learn about next week called concentration gradients. So the glucose will always flow in once the door is open, so to speak. So that phosphate can be transferred ADP to ATP, blah, 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 blah. And they're just showing you the thing. And it does all those things. We may need the energy to open the door for certain things to enter the cell. It may be, we use it all the time for our muscles to fire. When muscles fire, they change shape, or we need it to store energy in a bond, which we do frequently. That's chemical work, mechanical work. I never heard the term transport work, but we'll go with it. I like the way they put a lot of these in there. And that concludes that chapter. And I just, in the limited time we have left, 8 to 10 minutes we have left, I just want to introduce where we're going. This is the one that I sent you. The other, and you might recall when I was away on last Friday, I used this just as a little introduction because I wanted to do the proteins and the DNA RNA stuff in front of you because I think it's a little easier for me to give it greater context. I could read this to you if it's this is background. Okay, you had a lot of this stuff, I'm sure. I'm sure plenty of you are going to go over there and look at your bones. But go ahead, have fun. There's a party in there going on. So you can read cell theory, and I gave you the background on that, and all the diversity of cells, which is just brilliant. One thing that we'll focus on is we look at the different cells, and we particularly look at nerve and muscle cells. And I'll also talk a little bit mostly about bone and the uniqueness of bone vis-a-vis cellularly. You're going to see that the structure and the function are constantly of interest. Okay, the uniqueness, simple things, doesn't mean they are perform simple functions. Okay, they're important. Here, this makes sheets. Here, they're connected tissues. They're connected to one another, but they're not in sheets. They're not all next to each other. Here, they've lost a nucleus. It's just a vessel for carrying oxygen. Da-da-da-da. All those things. I did all that in that presentation I did for you. But there's a lot of diversity in the cells, and that accounts for a lot of their functionality. And in general, what we're going to see, the magic of the plasma membrane. We're membrane-bound organelles. It isn't just a plasma membrane. It's the membrane for the nucleus. It's the membrane for a mitochondria and other intracellular membranes like lysosomes, for instance. They are all the same. That's why I spent the time with phospholipids. If you take phospholipids and throw them into water, they form a membrane. You don't have to encourage them. That's their natural tendency. It's a bilayer. The fact that in our cells, that bilayer is able to becomes invested with proteins, and that's why I made that big deal about with the folding of the proteins. And I'll go and fold the papers again if you like, and all that stuff. I'm sure I wouldn't want to do that to you again. And so you can bury that into the membrane, and it can tether there, and the whole cholesterol that'll secure it there. That's the idea of these membranes, and what those proteins do in it, along with the cholesterol allowing it to open and close. We have a fluid mosaic model, effectively a membrane that by definition, and all living membranes are like this, they're called selectively permeable. Some stuff can get in and some stuff can get out. It can be the cell membrane, it can be the mitochondrial membrane, the nuclear membrane, it doesn't matter. If it's a membrane in us, it is by definition selectively permeable. There's got to be a way to get, whether it's nutrition in and waste products out in some way, shape, or form. Okay, so yes, it's a boundary. It doesn't necessarily mean it's only the cellular boundary. The cytoplasm, it's got the organelles, and it's a very interesting area because it has different chemicals. It's acidic and basic. It has a variety of salts going around, and inherently, what you will see, it's a relatively hostile environment. Okay, comparatively, there's so many chemical reactions going on, it can cause things like proteins and polypeptides to become unstable. Therefore, we have something that you'll see shortly, and we go into at length, called the endomembrane system. We have all of those organelles, the nucleus, the endoplasmic reticulum, the lysosomes, the Golgi complex, all the structures you learn about are all roadways covered and encased in membrane. Membrane protects, membrane isolates, compartmentalizes, all of these things. And yeah, the DNA is the control. This is what I mean. It's useful to see the complexity. When I was a kid and we learned about cells, they drew that stupid rectangle on the wall and put a circle in it. That's a cell. Some two-dimensional structure that, oh yeah, it's got a nucleus. That's interesting. Who cares? When's recess? It's fourth grade. Okay, here you get they're not all this complex, but we look at the complexity for differences, and that gives us an idea as to their function. So the endomembrane system starts here where it exits the nucleus, and all of these passageways made out of phospholipid bilayers with embedded proteins provide this interesting passageway, whether it's something you're going to spit out, stick in a membrane, or incorporate into some membrane-bound structure like a lysosome, for instance. That's there. And then the distinction in the fluids, those that are inside and those that are outside. They're very, very similar in their composition in the sense of, and we'll go into this, how much is dissolved in it, how much is solute, or how much is solvent stuff dissolved versus the total amount of water. But they're all different. Whether it's fluid between the cells, your plasma, cerebrospinal fluid, similar. But if we do a spinal tap or lumbar puncture and get a white blood cell and cerebrospinal fluid, that's a highly ominous indicator of something called spinal meningitis. And that can kill you very fast. Very, very fast. If that person isn't treated, as soon as you know that, there's a likelihood they'll either be severely damaged or will die. is bacterial meningitis. All these secretions, what makes them different? Not a whole lot. They're basically fluid. Is there extra protein? Saliva versus mucus. Okay, you learned about this in connective tissue, the matrix. Nitrogenated carbohydrates, structural material. Some can be solid, some can be liquid, some can be semi-solid. Cartilage is semi-solid, bone, it becomes solid as it absorbs calcium. So when you learn about, we do a whole unit on bone here with the physiology. Got nothing to do at 2 o'clock. Stick around here. I'm doing fractures today with my advanced anatomy class. One of my favorite talks. And so all of those things are there. And that's really what it is, the bilayer, the fluid mosaic. And what we're going to look at is the role of those proteins, whether they have sugar giving it identity, or whether it's a coating to reinforce it, or we attach things to it. And then we're going to look at the junction points, the cell adherence molecules, and that's really what we're going to cover as we move on to this. So we're going to start here. And I'll see everybody on Friday. Hopefully we'll be done with the first test material. Thanks again. Hey.